We often talk about aging as if it's a software bug we can patch, or a single switch hidden deep in our DNA that we just need to flip off. The hope is we'll edit bad genes and stop aging entirely. But from a purely biological standpoint, that logic falls apart. If living a perfectly healthy life is an absolute advantage, natural selection should have prioritized it. Over millions of years of rigorous trial and error, biology should have produced immortal organisms by now. Since that didn't happen, we are left with a strange mathematical reality. If natural selection allowed physical decline to persist across nearly every species on Earth, it means our eventual breakdown is likely the biological cost of our early survival. Evolutionary selection operates with high intensity during our reproductive years, but its influence fades as we age, leaving our eventual physical decline as an unavoidable biological blind spot. In 1957, evolutionary biologists proposed a concept to explain this called antagonistic pleiotropy. It is a complex term for a brutal biological principle. Evolution really only cares about keeping an organism alive long enough to reproduce. After that phase is over, what happens to the body is mostly irrelevant to the survival of the species. This diagram shows the exact mechanism of the theory. If a genetic mutation makes an animal stronger, larger, or more fertile when young, natural selection will favor it. It locks that gene into the population, even if it causes fatal diseases later in life. While this mathematical model explains why we degrade over time, finding the actual DNA variants that execute this trade-off in mammals has been difficult to prove. For decades, it remained mostly a theory. To map how lifespan actually works, researchers needed to empirically show that the biological advantages we enjoy in our youth are purchased at the direct expense of our old age. A 2026 study led by researchers including Ahrens and Williams provided the first high-resolution map of these biological trade-offs. They tracked a massive cohort of 6,438 genetically diverse mice throughout their entire lives to identify the specific genes driving these patterns. Most aging research simply looks at how long an animal lives and records the final time of death. This team used a different technique called actuarial mapping. Actuarial mapping works by analyzing the exact same genes across progressive, isolated age brackets. Instead of asking what a gene does over an entire lifespan, researchers asked what that gene does at 50 days old versus 500 days versus 1,000 days. They identified 29 distinct genetic locations, which they named vital loci, that shape mortality. But these loci do not operate on a static setting. The genetic code controlling our lifespan is a set of shifting variables. Their effects rewrite themselves depending on what stage of life the organism is currently experiencing. We can see exactly how this works on this chart, plotting a specific genetic variant across an age timeline. In the organism's youth, the gene's effect drops deep into a protective zone, actively lowering mortality. But as the timeline reaches late age, that exact same gene sharply pivots, driving up the organism's risk of death. The researchers also identified a separate set of 30 genetic locations they called soma loci. These govern a very specific physical trade-off. They found that genetic variants pushing an animal to develop a larger body mass for early survival consistently correlate with a shorter overall lifespan. Even more surprising is that these genetic trade-offs are strictly segregated by sex. A specific locus might offer strong biological protection to a female for 700 days, while that identical genetic sequence increases mortality in a male during that exact same time period. These dynamic, flipping loci provide the definitive evidence of antagonistic pleiotropy. We can now clearly see the biological contracts our bodies sign to survive youth. This data dismantles the idea of a universal fountain of youth pill. Because genetic effects actively flip depending on our age, a therapy designed to permanently turn a longevity gene on for life would lead to disastrous consequences once crossing into a new age bracket. Any future medicine targeting the aging process requires incredible precision. We will have to deploy interventions that target specific genetic loci only during exact, tightly defined windows of our lives. Furthermore, the strict segregation of these vita and soma loci means that every single anti-aging treatment developed in the future must be carefully tailored to an individual's biological sex. A drug that extends life for one sex may actively shorten it for another. We are not going to simply turn off aging. Extending the human health span requires us to carefully renegotiate the lethal genetic deals our bodies made millions of years ago.